Hello students, how are you all? I hope all you are doing very well. Today I am going to discuss about cell organelles of part 2. I have already discussed about part 1 of cell organelles. If you have not seen the previous part 1 video, then you must watch that video first and then coming to this video. Okay, first is endoplasmic reticulum. The nuclear envelope is directly connected to the extensive system of intracellular called the endoplasmic reticulum. Endoplasmic reticulum, it is mechanically supported by a network. It is a network of intermediate filaments called the nuclear lamina, a thin felt like mesh just beneath the inner nuclear membrane. It is nuclear membrane and inner nuclear membrane present endoplasmic reticulum. Many protein folds in endoplasmic reticulum. N-linked glycosylation begins in the endoplasmic reticulum. The smooth endoplasmic reticulum are two types. One is smooth endoplasmic reticulum and other is rough endoplasmic reticulum. This is the smooth endoplasmic reticulum, where endoplasmic reticulum is smooth and here ribosomes are absent, and it is the synthesis of fatty acid and phospholipids. And sarcoplasmic reticulum membrane of skeletal muscle cells have storage of calcium, and the another is rough endoplasmic reticulum. Here ribosomes are present, and endoplasmic reticulum non-muscle cells contain similar calcium pumps but in smaller quantities. Flattened membrane bound sacs is present called cisternae. It is a flattened structure, it is called cisternae. Then Golgi body, Golgi apparatus, also called Golgi complex or Golgi body. It is the membrane bound organelles of eukaryotic cells that is made up of series of flattened stacked pouches called cisternae. The Golgi apparatus is responsible for transporting, modifying, packaging proteins and lipids into vesicles for delivery to targeted destinations. It is located in the cytoplasm next to the endoplasmic reticulum. You can see this previous picture that in the next to the nuclear membrane Golgi body are present. It is located in the cytoplasm of next to the endoplasmic reticulum and near to the cell nucleus. While many types of cells contain only one or several Golgi apparatus, plant cells can contain hundreds. Then coming to the next slide, the apparatus has three primary compartments. One is known as generally as cis, medial and trans. These are three. The cis Golgi network and the trans Golgi network, which are made up of outermost cisternae and cis and trans faces, are structurally polarized. The cis face near to the region of the rough endoplasmic reticulum and the trans face near to the cell membrane. It is cell. If it is nucleus, then here cis face are present and trans face are present in the cell membrane. These two networks are responsible for the essential task of sorting proteins, lipids that are received at the cis face. Networks is responsible for the essential task of sorting. This is the cell, this is nucleus, and here Golgi apparatus are present. This is cis phase and this is trans phase, and the cis phase receive the protein sorting, lipids are received, and the trans phase release trans phase by the organelle. The cis phase membranes are generally thinner than the other. Then coming to the chloroplast. Chloroplasts are located predominantly in the mesophyll cells of leaves. This is the whole cell of the plant, and here it is the chloroplast present. This is the chloroplast in broadly structure. Chloroplasts are located predominantly in the mesophyll cells of leaves. Chloroplasts, which occur only in plants, some protozoa and green algae, harness solar energy to produce ATP by photosynthesis. The photosynthesis occur in this region and produce through the production of ATP and contain their own DNA. Here you can this chloroplast DNA and lost most of their own genomes and becomes heavily dependent on proteins that are encoded by genes in nucleus, synthesized in the cytosol and then imported into the organelle. Next, coming to the mitochondria. In eukaryotic cells, ATP that powers life processes is produced by specialized membrane enclosed energy converting organelles. These are two types, mitochondria and chloroplast. Chloroplast have already discussed, and then mitochondria. Mitochondria, which occur in virtually all cells of animals, plants and fungi, burn food molecule to produce ATP by oxidative phosphorylation. Contain their own circular DNA. Here in the matrix, circular DNA present and heavily dependent proteins that are encoded by genes in the nucleus, synthesized in the cytosol and then imported into the organelle. Mitochondria also double membrane structure. It has outer layer and inner layer matrix. It is the matrix region where the inner membrane enfolding and form crystal enfolding, and in that region a racket-like structure is present which is called cristae, where ATP synthesis occur. Here mitochondrial DNA is present. Then here it is also a mitochondria. You can also follow this picture. Here ATP synthesis occur. It is the matrix region, and cristae, it is the racket-like structure. Cristae, this is like racket-like structure. It is called cristae, and ribosomes present here. Granules, this is granules, and it is the inner membrane. It is the outer membrane, and here circular DNA is present. Then coming to the peroxisome. Peroxisomes are simple membrane bound vesicles contain oxidative enzymes. Peroxisomes, or microbodies as they are also called, are multifunctional organelles containing more than 50 enzymes. Here originated from endoplasmic reticulum. These organelles are named peroxisomes because they are the site of synthesis and degradation of hydrogen peroxide. That's why it is called peroxisome. And a highly reactive and toxic oxidizing agent present in this organelle. Hydrogen peroxide is produced by a number of peroxisomal enzymes including urate oxidase glycolate oxidase and amino acid oxidase that utilize molecular oxygen to oxidize their respective substrate. The hydrogen peroxide generated in this reaction is rapidly broken down by the enzymes catalase, which is present in high concentration in these organelles. Peroxisomes also present in plants. Plant seedling contain a specialized type of peroxisome called glyoxisome. This is the picture of peroxisome. Here it is the membrane transport protein present here, and this is the plasma membrane, and it is lipid bilayer, and hydrolytic enzymes are present there. Look at this picture. These are the membrane transport protein, that is glycosylated. Plasma, it is plasma membrane. Matrix, this is the matrix, and here hydrolytic enzymes are present. This is the structure of lysosome. Then coming to the, this is the structure of peroxisome. Then coming to the lysosome. Lysosomes are membrane enclosed organelles filled with soluble hydrolytic enzymes that digest macromolecules. Lysosome contain about 40 types of hydrolytic enzymes, including proteases, nucleases, glycosidases, lipases, phospholipases, phosphatases, and sulfatases. These are the enzymes which present in the lysosome. All are hydrolyses that work best at acidic pH. Here the condition is acidic optimal activity. They need to activated by proteolytic cleavage, which require an acid environment. The lysosome provides this acidity, maintaining an interior pH of about 4.5 to 5. The membrane of the lysosome keeps the digestive enzyme out of the cytosol. This is the cell membrane, and it engulfs the particles, and then gradually these particles degraded, and the degradation after releasing from the cell membrane. In this way, lysosome play a major role in the releasing of unwanted materials. These are the cell organelles. Pause your video and read very carefully. If this video is helpful to you, then please like, share with your friends, and comment if required any notes. Here, that's all. Coming to the next class with another video. Thank you.